to start with, we have two great postdocs from the CETPD at the University of Dundee, Maria Rodriguez-Rios and Connor Craigon, who will tell us all about a new platform called the BromoCatch. So Maria, before joining the Center for Protein Degradation, did her PhD with Mark Bradley at the University of Edinburgh, working on chemical probes for detecting proteases as biomarkers in inflammation. And Connor earned his PhD from the University of Dundee, working with Alessio Giuli on projects including the familiar Bromo tag. Maria and Connor, the floor is yours, and we look forward to hearing all about this new technology.
Great. Thank you so much for the kind introduction. And here we are. Connor and I will introduce you to BromoCatch, a new self-labeling protein tag platform for protein analysis and live imaging that we have developed here in the Giuli lab in collaboration with Tokris Biotechnie over the last couple of years. So what are self-labeling tags? They are small proteins or peptides that can be fused to a protein of interest and used in combination with a bivalent probe for analyzing proteins of interest. The specific probe will have an orthogonal functionality that allows you for imaging, immobilization, or bioconjugation of your target protein. There are examples of very successful SLPs in the scientific community, including HaloTag, SnapTag, or SpikeCatcher SpyTag. They are really powerful tools that have helped answering a lot of research questions so far, and they have been excellent. But there are some incompatibilities with the fusion to the protein of interest. Sometimes you have some immunogenicity problems, and the speed of labeling can pose some challenges with these platforms. So with these challenges in mind, we came up with BromoCatch. We kept these challenges in mind when designing and developing this platform. That we hope it becomes a useful tool for the community and helps address some of the current challenges. So the fusion tag in BromoCatch is a bromodomain protein. And bromodomain proteins are proteins that have been widely studied in the Giuli lab for quite some time now. And they have been, they bind acetylated histones in the cell, and they regulate transcription. And back in 2014, the Giuli lab tried to understand the function of individual bromodomains by engineering the specificity of an inhibitor using the bump and hole approach. This bump and hole approach takes a pan-selective bromodomain binder and installs a bump on it that will prevent binding to the wild-type protein. And complementarily, you take a bromodomain of interest and you make a mutant where you mutate the binding pocket with a hole that will accommodate that bump that you install on the inhibitor selectively. So with this approach, you engineer selectivity for your mutant, and this approach allowed the Giuli lab at the time to understand the function of individual bromodomains specifically. The same approach was used later in 2021 to develop BromoTag, a Degradation system that was developed here, and it uses a bump and hole mutant that in this case is functionally silent, and it can be used to fuse to a protein of interest and induce degradation of that protein in combination with a bumped PROTAC that has a VHL ligand that can hijack the degradation machinery and induce degradation of the target. This system has been commercialized by Tokris Biotechnie recently and has been used successfully in the TPD field by different labs. And I think it's a great technology, but we were thinking that we could expand the possibilities of this platform beyond degradation by going covalent. So we could engineer that covalency by incorporating a cysteine in the binding pocket and an electrophile in the ligand, and that would open the door to many more applications, including imaging or protein purification, among others. Additionally, we looked at the size of the tag, and we shortened it to make it to get it to a minimal size of 13 kilodaltons by removing a non-necessary region of the protein. And we also designed a library of BromoCatch-specific probes that could be used for different applications to show the utility of our platform for fluorescence imaging, biotin-based assays, bioconjugation, or TPD. How we redesigned the system from BromoTag is we looked back at the bump and hole crystal structure, and we wanted to keep the bump and hole system with the mutation to make the hole that uses a leucine mutation to an alanine that goes from a big leucine residue into a smaller one. And on the inhibitor, as you've seen before, is an ethyl bump that comes out into that pocket. That allows us to keep the orthogonality and over the wild type system and allow for the specificity for the BromoCatch. For the BromoTag. And now going to BromoCatch, we wanted to look at positions in the binding pocket where we could look at residues that we could mutate to cysteines. And we identified methionine 442 and glutamic acid 438 that were both residues that oriented in close proximity to the aromatic ring in the ligand and could be potential sites to mutate into cysteine while incorporating the electrophile around this ring. So we modeled the cysteines and we looked at the distances that we would have between the cysteine and the aromatic ring, and we determined that we had 6 angstroms. So that is ideal to be able to install an electrophile in there that could potentially engage covalently with that engineered cysteine. And that was the case very similar for the methionine mutant we designed. So we took these two protein designs as potential tags, and we went on to look at the inhibitor more closely. And again, we wanted to keep the bump for the bump and hole and the orthogonality over the wild type. And we looked at the meta and para position of the aromatic ring as the positions where we wanted to include the electrophile. And at the time we were designing this library, some work by the Dana-Farber came out on how this was possible, engineering electrophiles around these positions, and that was really encouraging. And we kept designing our library with a different variety of electrophiles in both positions that could cover a range of chemical space and also reactivity. Before going into testing this in the lab or making the compounds and the proteins, we wanted to model the system in silico to assess its feasibility first. And for that, we did some covalent docking studies where we did model the system in Schrodinger, and we took the proteins and we took the ligands and we docked them covalently. And we were quite happy to see that we could maintain, the binding mode was maintained. We had great docking scores with low binding energies, and the covalent bond was generated for most of the ligands we screened. So that was really encouraging. So then we went on and tried we express the proteins, we synthesize our library of ligands. And the first question we wanted to ask is whether we had covalency and if any of these ligands could actually modify our protein covalently. And for that, we used intact MS that allows us to analyze the exact mass of a protein. And it's a useful technique to screen for covalent modifications of proteins of interest. So in this case, we ran samples where we had just our protein or the protein in presence of the ligand, and we looked for changes in mass that would indicate there was a covalent modification. And we did that with the whole library. We incorporated some controls as well, like the reversible binder ethylbam JQ1, some non-really reactive electrophiles. And we also incorporated proteins such as the wild-type bromodomain 4BD2, the bump and hole reversible mutant, as well as the 2 cysteine-containing mutants that we designed. And we were really happy to see that actually we got a few hits for the methionine to cysteine mutant, but we got a lot of hits for the glutamic acid to cysteine that seemed to be highly reactive. And also on top of that, we had no labeling of the wild type or the bump and hole mutant that is the reversible bump and hole system. And that was showing the specificity and minor off-target for any of these 2 proteins. That was really reassuring. And you can see here how one of these results looks like. You have the protein in absence of any ligand, or the protein incubated with the ligand, and you have a change of mass that corresponds to the molecular weight of that ligand, which is the acrylamide. And you also can see on the UV the shift of retention time. And that was the case for many of the ligands. We got complete conversion within 2 hours for many of them. So we were really encouraged by this. And we moved on into testing the library to understand the relative binding affinity of the library components for our proteins. And for that, we use differential scanning fluorimetry and assess the stabilization effect that the different ligands had for our proteins. So how this works is, many of you will be familiar with it. You have a protein that you can denaturate and determine the melting temperature. And generally, when a protein binds a ligand, there is a stabilization effect that results in a shift on that melting temperature. And that's why we assessed for our library in presence of the proteins that were the same design as for the intact MS screen. And we were quite pleased with the results because we could see that we had minor stabilization of the wild type protein, indicating that the bump clashes and prevents reversible binding, improved binding to the bump and hole mutant with the L2A mutation. And then it was the striking result was how much stabilization we got for the ones that were covalent. That was also confirmed by the intact MS. So ligands like the acrylamide were giving up to 26 degrees for the glutamic acid to cysteine mutant. That was really, really encouraging. So we had these results now from the intact and the DSF, and we already chose the BromoCatch protein as the ideal protein because it was more reactive in both of the assays we did. And on the ligand side, we went for the acrylamide para because it showed the highest stabilization. It had optimal reactivity and fast kinetics, and it's also a really well-established acrylamide warhead. So the acrylamide warhead is a really well-established warhead in the medchem field. So we also tried to do some co-crystals with the BromoCatch protein and ligand to be able to resolve see if we could see the covalent bond at a molecular level. And so we tried to crystallize the BromoCatch protein with our ligand, and we had some issues with the BromoCatch protein. So we had to move into a highly homologous BRD2 mutant that allowed us to do those co-crystals quite a lot easier. And we could see how the binding mode was actually maintained with an RMSD relative to the reference crystal under 1 Angstrom. And we could also see the continuous density around that bond really well on the confirming that covalency. And interestingly, thanks to the high resolution of our crystal, we could fit 2 conformations of the bond on that density, indicating that cysteine is pretty flexible. So with that, we have a ligand and a protein pair that seems to be working quite well in vitro and shows a lot of promise, but we haven't tested anything in cells yet. So this is where Connor comes in and explains a little bit of the biology.
Okay, thank you, Maria. Yeah, as Maria's obviously talked about, so far, essentially, we've developed this BromoCatch domain, which is this glutamic acid to cysteine modification, and we've essentially established in vitro this acrylamide best-in-class electrophilic warhead that can covalently bind in vitro. But what we haven't established is whether or not we can demonstrate this effect in live cells. So to begin with, just for determining potent binding of our acrylamide warhead to our BromoCatch domain in cells, we decided to develop a nanobret assay. So what we first did is we generated this DNA construct where we have a PMCV-driven nanoluciferase tethered to our BromoCatch tag. And in essence, at the same time, we synthesized an ethyl JQ1 body P tracer. And the way a nanobret works, in essence, is that when you have the nanoluciferase expressed tethered to your BromoCatch domain, you add in the fumarate substrate, and this induces luminescent output. This luminescent output stimulates the fluorophore on the body P and produces a secondary Brett emission that we can detect on a spectrophotometer. So the way we set up this assay, we did a pre-incubation of our nanoluciferase tethered BromoCatch domain with a 1 micromolar treatment of our ethyl JQ1 body P tracer. We then subsequently titrate in our electrophilic warhead at various concentrations. And what we're essentially looking for is competitive displacement of the tracer, which would result in a reduction in Barrett signal that we can detect. And so when we did this, we actually did a 10-minute incubation step with the ultraphilic warhead before we actually looked at the Barrett signal. And this is the outcome of that experiment. So when we do our ethyl JQ1 OME and reversible inhibitor in this system, we get an IC50 of 88 nanomolar. But very interestingly for us, what we actually demonstrate is that when we do with our ultraphilic warhead MR116, we do in fact get a slight reduction in the IC50, so slightly more potent at 31 nanomolar. And we conclude from this that we can get potent binding of our acrylamide warhead to our BromoCatch domain in live cells. And also, just to reiterate there, we did a 10-minute incubation, suggesting this is quite a rapid uptake of this interaction. So the next real question we want to ask then is, well, we can certainly show that we can have live cell binding quite potently in live cells, but can we actually show covalency in live cells? So to do this, what we essentially did was we inversed our nanobrett assay from the previous experiment and utilized it as a resonance time assay. And so the way this assay works, in essence, is instead of starting with your ethyl JQ1 body P tracer and then adding in your electrophilic warhead, what we do is we used a set concentration of our electrophilic warhead, incubated for 2 hours, and then subsequently added in a saturating concentration of our ethyl JQ1 body P tracer. And what essentially we're looking for here is that if the warhead has formed a covalent bond, it will not be able to be displaced by our ethyl JQ1 body P tracer. And so we should not see during the lifetime of the experiment any increase in Barrett signal from our MR116. So when we do the experiment, we use DMSO as our vehicle control, and we used ethyl JQ1 OME as our inhibitor, our positive control inhibitor. And what we can see here is the outcome of a 2-hour experiment. So when we do with DMSO and ethyl JQ1 OME, we can see a rapid increase in Barrett signal that sort of plateaus at about 30 minutes, which is what we expect. But very important for us, what we do see when we treat with MR116 pre-incubated at 250 nanomolar for 2 hours and then subsequently treated with our tracer, 25 micromolar, is we do not see any increase in Barrett signal in this assay, which we come to conclude that we can actually see the covalent modification of our acrylamide to our BromoCatch domain. So at this point in the story, what we really have is we have this BromoCatch domain tag, and we also have this acrylamide warhead that we've shown in vitro and in cellular we can cause the formation of a covalent bond. So now the real big question is, can we functionalize the acrylamide so that we have a bunch of utilizable probes for doing various different types of protein manipulation and live cell imaging in living cells? So a common use of these sort of SLP tags is sort of a biotin-based protein analysis. So one of our first experiments into functionalizing our acrylamide warhead is the development of a biotin functionalized bivalent probe, which we're calling MR169 here. And then essentially what we want to do is validate this both in vitro and in cellular. So in order to do in vitro validation, we took the intact mass spec analysis that we'd done for previous experiments earlier. We used our purified BromoCatch protein, incubated it with MR169, and then subsequently did the analysis. And what we were able to demonstrate with that is that we can get complete covalent modification of our BromoCatch purified protein with our MR169 when it's treated at a 2-to-1 ratio of the MR169 to BromoCatch protein. So we have in vitro evidence of the covalent modification. And then now we want to show that we could actually use this to identify biotin-related proteins of interest in cellulose. So to do that, we actually developed a new DNA construct where we have a PCMV-driven H2B tethered to our BromoCatch domain. And then we express this in HEC293 FT cells transiently. And then we did a subsequent titration of our MR169 in those HEC FT transfected cells, and then performed a Western blot to see if we could identify any biotin-related protein. And what we actually see from this, if you look at the Western blot here, is that we can see a concentration-dependent increase in biotinylated H2B BromoCatch protein only in the transfected cells. That was our first observation, which is great for us. And secondly, what we also noticed is that we don't get any off-target biotinylation occurring either in the wild type or in the transiently transfected cells. And what this really suggests to us is that our probe is both effective against our BromoCatch domain, but also that it's selective for the BromoCatch system. And so we think this will be a very useful probe tool for people who are interested in protein purification or pull down experiments. Another common utility in these SOP systems is the ability to recruit an E3 ligase that can facilitate degradation of a tagged protein of interest. So in that sort of vein of logic, we decided to develop and synthesize our own version of an E3 recruiting degrader. And so this is our acrylamide warhead tethered to PEG linker and then subsequently to a VHL E3 ligase binder. And so now that we have this probe in hand, we really want to validate this in cellulose. So previously we did a lot of transient systems, but because we're looking at degradation using a covalent degrader, we felt that we would rather have a more physiologically relevant level of expression. So we actually decided to do an endogenous knock-in system. So this construct here on the right is the construct we designed and implemented into HEC293 cells. So we use CRISPR to tag in this EGFP IRES high-bit BromoCatch knock-in to the N-terminus of BRD4. We did some EGFP mediated reporter selection for cell lines that had the GFP expression. And then subsequently, once we grew up the clones, we were able to establish some early selection for those that had the knock-in through our high-bit lytic assay looking for high-bit signal. So with that in mind, we now have a model system which we can probe for degradation, and we also have this newly synthesized probe. So the next thing we did was we did a high-bit live cell kinetic assay, which essentially is we take that model system that we designed and developed, and we add in a large bit construct that allows the expression of this large bit protein. When this comes into proximity with the high bit, it forms an active nanoluciferase, which can be used as an indirect mechanism for looking at protein levels of the protein that's tethered to the high bit. So in this case, BRD4. And so what we ultimately did then was we did a 9-hour incubation with a fixed concentration of our compound, in this case, MR170 at 2.5 micromolar. And we just observed whether or not we could see loss of protein expression, loss of protein level over time. And what we observed actually was that we could get about a 60% reduction in protein levels over a 9-hour period with a t-half of about 84 minutes with our MR170 covalent degrader. And this is not untypical of the sort of covalent degraders in terms of their activity, since it's no longer acting as a catalytic in a catalytic manner. But at this point in the project, we really had never actually looked at whether or not the original tags design was fundamentally based on the BromoTag system. And so we had never actually thought to check whether or not we could actually use AGB1 in this system and it would function as a catalytic reversible degrader for this technology. And as it turns out, when we do treat in the same assay with AGB1, we do find that we can still actively degrade our target very potently and reverse using this reversible PROTAC. And so this suggests two different ideas really, which is that our BromoCatch being compatible with AGB1 means that we can use our BromoCatch system as a degrader system, a degron system, much like the original BromoTag, as well as we're using AGB1 as a multiplexing tool with other probes we've developed using BromoCatch to probe protein dynamics in living cells. So one of the most common approaches in SLPs fundamentally is live cell imaging, where you're looking at tracking proteins of interest that have been tethered with this covalent tag. And our funders in the lab are Tokris Biotechny, and they have extensive experience using these Janelia fluor dyes for live cell imaging. So what makes these probes so useful for live cell imaging fundamentally is that they are these rhodamine based, they're cell permeable, and they're relatively small. But the most important aspect about them that makes them very useful for as probes for live cell imaging is that they have this unique ability where they are fluorogenic switch on probes, meaning that only when they're bound to their target do they actually change shape and become fluorescent. And this is very useful for live cell imaging in particular because it means that you don't require lots of washing steps or you have very low background signal in the end. So with that in mind and with their expertise and help, we were able to develop the C10852S probe, which is a BromoCatch probe with a Genelia 4635 dye on it. Initially, we did some in vitro analysis just to validate that we could get covalent binding to occur. So we did use purified protein and we incubated with C10852S inside cells, and then we observed whether or not we could see this covalent modification. And in fact, the only one of the 3 purified proteins that we used, the only one that was capable of doing the covalent modification was the BromoCatch tag. However, there's a known effect where this Genelia fluors can actually be activated in the presence of SDS micelles. So we next did another in vitro analysis where we just looked at the purified protein in HEPAS buffer just to see whether or not we could get sort of a native switch on of the Genelia fluor probe in the absence of SDS. And in fact, what we did notice is the highest level of switch on occurred when it was bound to the BromoCatch purified protein. So obviously, with all that, the big question then is whether or not we can actually use this probe for live cell imaging. So what we did is we took the H2B DNA construct from earlier, which contains our BromoCatch tag, and we transfected them into U2OS cells. And then from there, we then did a subsequent treatment with C10852S at 100 nanomolars for 8 hours. And then we used a counter stain of horse 33342 since H2B is a nuclear protein. And we just wanted to observe whether or not we could see this Genelia switch on when bound to the BromoCatch domain. So obviously with the DMSO, we don't see any background in the Genelia fluor channel, and we obviously see very bright nuclear staining of the horse 3342. But when we do it with the C10852S, we actually do see very bright nuclear staining and switch on of the Genelia fluor only when we're treating with our C10852S. Now, obviously, the next big question for us is, well, do we see any background signal when we treat non-transfected cells with our compound? And actually, in fact, what we see, we don't actually see any switch on in this background U2OS cells, which is promising, suggests that our Genelia fluor switch on is selective for a BromoCatch domain. And just to bring the point even further, we even did a co-treatment experiment where we're competing against it with the ethyl JQ1 OME inhibitor, blocking the active site of the BromoCatch domain. And actually, we don't see any background switch on as well. So we think with this, we have a selective and effective probe that can be used for live cell imaging. And so just to reiterate what we've said today, we have developed this new self-labeling protein tag platform for manipulation of proteins in live cells and live cell imaging. And we've developed a library of probes that have different functionalities like biotin, E3 ligase recruitment, and fluorescent activity for microscopy, and that these will actually be developed very soon, commercialized very soon with our collaborators, Tokers Biotechny. I just want to acknowledge some people. So I would like to acknowledge the BromoCatch team here at CETPD, as well as our counterparts at Tokers' labs down south. I'd also like to thank the University of Dundee, our funders, Tokers Biotechny, and the Dana-Farber Institute for having us today. Thank you, and any questions?